Hi, my name is Mira Han. I'm from University of Nevada, Las Vegas, and I'm going to tell you a little bit about our experience trying to train machine learning on enhancer perturbation data. So understanding how a particular enhancer finds a specific target gene to enhance its transcription is still an open problem. And we thought maybe we can learn such rules from data through machine learning. And the motivation for it came from these first three studies that were published in around 2019, 2020. There were many studies that used CRISPR interference to perturb hundreds of enhancers and read out the gene expression results in multiple genes. And we thought it's enough data to use a supervised approach. And the largest data set to date on enhancer perturbation is this study, the Gasparini et al. From 2019, where they used multiplexing of guide RNAs across hundreds of thousands of cells and looked at the gene expression readout through single-cell RNA-seq. And we're using this data set as our main training data set. And the prediction is based on this gene expression downregulation that they call positive. And based on their study, there were only about 600 genes that enhancer promoter relationships that were called positive out of about 66,000 enhancer promoter pairs. So that's less than 1% positive relationship. So it was an extremely imbalanced data set. Before we get to our model, this is the existing model that's kind of accepted in the field. And it is a very simple but elegant model that contains two parts. One is the enhancer activity, which is measured by the histone modification, H3K27 acylation, and the DHS peaks. And then the other component is the intensity of the high-C contact between the window containing the enhancer and the promoter of the gene. In contrast to that unsupervised model, the model that we were training on the perturbation data set is a supervised gradient-boosted tree using XGBoost. And the features that we include these variables that go into the ABC model, but additionally we have some other histone modification data and about 250 transcription factor peaks from ChIP-seq on the same K562 cell line using the ENCODE data. And we set aside chromosome 5, 10, 15, and 20 from Gasparini as our test data set. And then we used the two additional studies we introduced earlier as independent test data set. So here are our results. We generally found that once you do supervised learning with additional transcription factor information, you do see increased prediction accuracy. And that was in both the outer cross-validation evaluation and the test chromosomes that we set aside. So the XGB are our models compared to the ABC model and then the baseline distance-based model. And if you include all the transcription factor information, that's the full model, then you see consistently about 6% higher precision recall compared to the model that excludes all the transcription factor information. And you do see increase against the ABC model as well. So this was based on the test data set aside from the same Gasparini data set. But when we applied it to the additional two independent data, the Fulco et al. And Schrei-Vogel et al., we didn't see the same increase in performance compared to the ABC model. And so we thought, what's the difference between these two data sets and the other earlier Gasparini? And the main difference is that these two data sets were targeted experiments. So Fulco only read out about 20 genes, Schrei-Vogel only read out about 200 genes. So, no, I think I said it wrong, 30 genes, 200 genes. They justifiably focused on genes that potentially have a positive enhancer regulating that gene. So we went back to the Gasparini et al. Data and tried to filter it in a similar way. So this time we only included genes that have at least one positive enhancer regulating that gene in the data. And overall, you see a significant increase in performance, but again, the overall performance of the XGBoost model, including transcription factors, are not very different from the ABC model. So our interpretation is that if you already know that there's a large effect enhancer regulating your gene, then it seems like the H327 acetylation and high-C contact is all you need to find that enhancer. And having additional transcription factor information doesn't really give you much edge. We looked at some feature importance of our model to understand what factors are important. And generally, we found that ABC model largely captures the most important factors in enhancer-promoter relationship. So the high-C contact was the most important factor in predicting positive enhancer-promoter pairs. And then H3K27 acetylation at the enhancer was also ranked very highly. The way that you read these kind of graphs is, so the further you are to the right, the more positive prediction. The further you are to the left, the more negative prediction. And then red means high feature value, blue means low feature value. So when you see a red in high-C contact, that means higher the high-C contact, the more positive you're going to predict the enhancer-promoter functional relationship. And then you can see that here as well. So the high-C contact and the SHAP values generally have a positive relationship, meaning that the stronger the high-C contact, the more positive your prediction. And you can kind of see that the high-C contact doesn't really need to be very strong. By the time you're about 0.005, you're already predicting positive. But in addition to corroborating the ABC model, we also found some novel insights regarding enhancer-promoter relationships. What we found was that our model can predict the negatives much better than the ABC model. And one of the reasons was that it was picking up signals predicting genes that are insensitive to distal enhancers. So here we're showing you these patterns where the blue value, the stronger that value, the more negative. So the blue being on the right side means that if you have strong histone H327 acetylation on the promoter of the gene, then you're more likely to be predicted negative, meaning that that gene is not likely to have a functional distal enhancer. And there are a couple of transcription factors that are also increasing that prediction, and these were generally correlated in their SHAP values. And that's kind of the reason why you see higher performance for our XGBoost model compared to the ABC model. You see that predicting positives are generally not very different between the two models. But if you look at the false positives, we have a much reduced false positive rate because we're able to predict the insensitive genes. Now going to the features that are important for positive EP relationships, we found that several transcription clusters are actually predictive for positive relationships, and we're just highlighting a few of those NMF clusters. So for the transcription factors on the promoter side, you see general RNA polymerases showing up, and the RBFOX2, which is a splicing factor. And for the enhancer side, you find proteins that are generally known to regulate cell cycles. So this is a K562 cancer cell line. So cell cycle genes, hematopoiesis genes like the STATs, the GATAs, they're showing up in the enhancer binding sites. And then there was an interesting feature for transcription factor clusters that actually predicted negative enhancer-promoter relationships. And these were curious because they were well-known activator proteins that are part of the AP1 complex. And those are known positive regulators. And so the interpretation on our side is AP1 complex is a known protein complex that is known to bind enhancer hubs. They're enriched on enhancers that multiple enhancers together regulate a protein gene. And so we think that what it's picking up is the cases of robust enhancer hubs where CRISPR interference targeting a single enhancer doesn't show any effect in the downregulation of the gene expression. The last feature that I wanted to talk about was the density of the enhancer and the promoter in the region of the gene was an important factor that showed up in the higher rankings. And you can actually see that in the raw data itself. If you go back to the Gasparini data and sort their positive EP pairs based on density of how many enhancers, how many promoters are in that region, you find that if you go to regions where there are lots of enhancers and lots of promoters, you need a much higher high-C contact for that pair to be called positive. And you can see that our machine learning picks it up as well, because generally there's a negative relationship between the SHAP values and the density of the region. If you are in a region where there are lots of enhancers and lots of promoters, then your EP pair is less likely to be predicted as positive. But we saw that there were, once you focus on the enhancers that have no strong contact with the promoter, there were a very few subset of positive cases that showed up that were actually showing the opposite pattern, meaning that you were more likely to be called positive only when you were in a very high density region. And we went back to the raw data to pick those positive cases and look at the characteristics. And what we found was that generally these enhancers didn't have the regular characteristics that make an enhancer positive. So generally we think that strong high-C contact and very high enhancer activity is what makes an enhancer positive. But these enhancers were generally very low in contact with the promoter and had relatively lower H3K27 acetylation. But they were found in regions where there were very high number of promoters and enhancers. So in summary, we showed that integrating transcription factor information in the supervised model increased the accuracy of predicting enhancer promoter specificity. And the better performance was achieved by learning where the CRISPR interference experiments didn't work. And it wasn't that the experiments were wrong or anything, it was that it was picking up robust transcription activation, either on the promoter side or the enhancer side, where it acted against the CRISPR interference experiments. We identified atypical enhancers in high-density regions that do not follow the characteristics of positive enhancers. So it seems like there's more to learn about this relationship, but it'll be in the enhancers that have modest effects or enhancers with redundancy or synergistic effects in their regulation. That's all. These are my students who were participating in the study, and this is Dylan, who's the lead author of the study, and you can find the paper in bioinformatics.
Thank you for the nice talk. So two things. One is a comment on the last bit where you're talking about the like these type of enhancers. I think this goes along well with the promoters acting as enhancers of other genes, where those wouldn't have maybe as much K27. They would have the promoter markers, and then they would act through like lower distances. So I mean, strong versus weak high-C contact, that depends on how you are normalizing, but that may make sense. So related question is, a lot of these Gasparini et al. And all these other validation experiments that you used, they, a lot of the enhancers that are validated are very short distance from the gene target, right? And then like it's really important how we analyze the high-C data or the contact. They may be within the same bin. So how are you handling like those very short range distances and how the high-C data is processed in that?
Yes, so In the Hi-C matrix itself, if they are in the same window, that window also has a value for Hi-C. So we were just taking that value on the diagonal of the Hi-C matrix as that value for the enhancer promoter pairs that are in the same window. But we do have distance as part of our features as well, and that's basically the second feature. Second feature that shows up as
it's a well-known issue with ABC that it mainly captures the distance in a very convoluted way through Hi-C contacts. The self-interaction, those values are going to be highly confounded by how many restriction enzyme cut sites are within that bin and so on and so forth. So using them as they are can be a little bit tricky. For short-range distances, like Hi-C data can be very tricky to analyze or try to make sense of it. Thank you. So.
A really interesting talk. Thanks. So, on one hand, integrating transcription factor peaks at the scale you're doing is only possible in a couple of different cell types where you have coverage. But on the other, the types of TFs that you're identifying are broadly expressed and fairly consistent in binding across cell types, some of them at least. So are there more general rules you can pull out or general advice if we're trying to predict enhancer promoter interactions in very different cell types?
What we found was that other than that AP1 complex, generally, if there were a transcription factor found in the enhancer, it was predicting the positive side. So it didn't seem like what transcription factor it is not very important. As long as there were transcription factors binding, it was positive. So maybe that gives some hint, I'm not sure.
Is it the accessibility? Could it just be that promoter is
So that sort of predicts constitutive promoter activity or something like that? Oh. Okay. Thank you.
One more question and then we move on.
Really great talk, thank you. For the Gasparini paper, that was on K562, correct? Yes. Have you tried out, I know probably for other cell types it would be much, much smaller screens, but are there screens in other cell types that you ran this model on and then compared, positive EPs? And, I'm really curious, especially about those atypical enhancers.
I see. No, we didn't. The only independent tests we did were FOCO and Trivoga, which are both also K562. But we know since then there are many other data sets that have been published. We'll probably see lower performance, but it will be interesting to take a look, yes.
Thank you.
Thank you. Thanks.